So with a wide range of viral vector-based drugs already approved, viral vectors are expected to remain the primary delivery mechanism for the foreseeable future. However, as the demand for viral vectors increases, addressing the challenges related to their manufacture and scale up is critical. And so before we dive into our discussion, I want to give the floor to both Dave and Curran for just a little bit, for a brief moment to share with you uh, the vectors their respective companies utilize and why. And so, Dave, I'd like to start with you. Tell us about the vectors that Candel Therapeutics is using to date and the how and the why behind it.
Yeah, thanks, Aaron. So, I mean, it's great to be here, and I really appreciate the invite. In terms of my current areas of concentration, my company, Candel Therapeutics, focuses on both adenovirus as well as herpes simplex virus-based platforms. Generally speaking, the use of these particular vectors is based on things such as payload capacities, cell tropism requirements, in these particular cases, their ability to safely deliver their respective payloads to the tumor microenvironments in an in vivo situation. Over the years, I've worked with adenovirus, lentivirus, as well as retrovirus and some others. So my hope here today is that I'm able to provide at least some useful information to a few of your listeners. But again, thank you for having me today.
Oh, thank you for your time, and I do look forward to hearing all the insight you have to share with us, given certainly your robust background. Curran, I want to offer the same opportunity to you to share with us what REGENXBIO is doing in the AAV space and how you're utilizing the vector.
Sure. Our company is steeped in what's called the NAV technology, which is AAV-based. And largely our programs are based on either AAV8 or AAV9, which are programs that originate all the way back to work done early in Jim Wilson's lab at UPenn. We've taken them a long way from there. AAV9, for example, is used in CNS indications. MPS 1 and 2 are two programs that we have in MPS 2 is in pivotal studies right now using AAV9. And then AAV8 is used both in our RGX-314 ocular program and also in our Duchenne program, which is a systemic administration.
Wonderful. Thank you. Okay, and we'll be getting deeper into that as we go on through our conversation. But first, Dave, I'd like to start with you. I want to talk a little bit through quality control assays as they are performed on viral vectors. You know, what are they, and kind of if you could kind of set the stage for us. I know it's very broad, but if you can kind of set the stage for us with regard to quality control assays.
Sure. Well, I mean, there are dozens of different types, as you might imagine, analytical methods that are used. Some are broad ranging, I think, not necessarily specific to your vector type. Some of the common ones that a lot of folks on this call would be aware of in any event, things like host cell proteins and host cell DNA and other what I call process-related impurities. There are methods, obviously, to quantify various aspects of things like that. There are QC methods designed specifically for product-related impurities. I'm sure Curran will speak to things like empty capsids with respect to AAV. But I think the major focus, or certainly where a large amount of energy is spent in this space, is really on coming up with a compelling package of demonstrating potency of your product. And generally speaking, we're talking about in vitro potency is a big area of focus for both the agencies as well as companies. There can be a number of different methods to try and do this. As a matter of fact, oftentimes it takes a number of different methods to try and demonstrate this. Some are very complex cell-based methods and whatnot. But really, you know, that area in particular, within this sort of broad-ranging area of QC methods, I really think that the potency family of methods continue to provide the most significant challenges to sponsor companies.
Absolutely. And Curran, I want to, of course, offer you the same opportunity to speak from an AAV perspective. From a quality control, what are the quality control assays performed on viral vectors?
Well, I think a lot of them originated through biologics in terms of how we think about viral vectors, but then we get into sort of the 3D science associated with vectors, and it starts to add, new challenges to the analytical format. One of the ones that we use in process that's interesting and relevant to gene therapy is, as mentioned earlier, measurement of empty and full capsids, which you can do using methods that could take 2 days to turn around, like ultracentrifugation. But we've come up with methodologies and shared those publicly around just using absorption ratio A280 to 260, which allows you to look roughly at the amount of empty and full capsid in process where you're maybe making a delicate separation. So we've had to kind of modify, what we learned from biologics as we went into gene therapy. The other one I would say that's emerging is the measurement of full genome. And so when you package the capsid, you may not package 100% full genome of the target transgene. And so looking at the DNA analysis that's associated with that, to know that the product is primarily full genome that's been packaged is becoming more important.